Okay. Hi class. Moving on to our lecture on the nervous system. This is a complex lecture, so you might have to um, go over the videos multiple times. And at any point, if you think it's not clear or if you are not sure about something, please feel free to email me. Okay. Now, to start with, I want you to know that every single cell of your body is set up like a battery. Now, if you look at the picture of the battery on the right hand side, what do you notice about the battery? You are right. You see a positive end and a negative end, right? So, in a battery, we have separation of charge. Do you agree? We have a positive end and a negative end, so there is separation of charge. Okay. If you look at a human cell, the plasma membrane of a cell also helps in separation of charge. Now we know that the plasma membrane separates chemicals, right? It separates the intracellular material from extracellular environment. But not only that, I want you to know that the plasma membrane helps in separation of charge as well. In the picture on the right hand side, top right, we see a plasma membrane. Embedded within the plasma membrane are different types of channels. We are not talking about the channels right now, but what we need to understand, if you look inside the membrane, you see a bunch of negative signs in black, and outside you see a bunch of positive signs indicated by the green balls. Do you see that? So my point is, when a cell is at rest, meaning it's in a non-excitable state, at rest or in non-excited state, a cell has negative charge inside and comparatively it's more positive outside. And this has been demonstrated by experiments. So if you look at the picture again on the top right, you will see that a needle is inserted into the intracellular membrane right here. And the needle is hooked to a computer. And with the needle, it can be detected that the inside of the membrane is more negative as compared to outside. And this difference of charge across the plasma membrane is called resting membrane potential. Do not let these terms bully you to submission. No. Resting membrane potential simply means separation of charge across the membrane. Okay? Okay. And resting membrane potential is always expressed by its value inside the cell relative to extracellular environment. Now, students often ask me, Rupa, how negative is the inside? Well, my answer is, it depends on which type of cell we are looking at. It is very different in neurons versus muscle cells versus neurons versus skeletal muscles versus cardiac muscles. So depending on which cell we are looking at, we will get a different value for the resting membrane potential. However, scientists take an average value, and the average value lies between -70 to -80 millivolts. I want my students to know that this is just an average number of resting membrane potential. So hopefully you understand that resting membrane potential is nothing but the difference in charge across a cell membrane in a non-excitable state when the cell is at rest, right? Okay, good. Moving on to another important, very important concept. Now, in order to appreciate resting membrane potential, we must understand the difference between intracellular and extracellular environment. Now, the question is, what do we find inside the cell versus what do we see outside the cell? Well, know that we have electrolytes inside the cell, right? And the electrolytes have charges. Now, by electrolytes, I mean ions. So we have sodium, potassium, chloride, calcium, all these ions inside the cell. Now tell me, do we also find these ions in the extracellular environment, meaning outside the cell? The answer is yes. But remember, their concentrations are very different inside as compared to outside. Okay? Now we will look at the chart. On the left-hand column, we see different ions. We see sodium, potassium, chloride, and calcium. And other two columns tell us the intracellular concentration of each ion and then extracellular concentration of each ion. Let's take sodium, for example. We do notice that sodium is more abundant in the extracellular environment. Chloride, same thing. It's more abundant in the extracellular environment, and same is true for calcium. We have more calcium outside the cell. The only exception is potassium. We have more potassium inside the cell as compared to outside. You, my students, my lovely students, you are responsible for knowing which... Rian, quiet. Shh, quiet. No. You are sorry about that. You are responsible for knowing which ion is in higher concentration inside versus outside. Do you need to remember the numbers? Of course not. Okay, so let's keep it simple. Inside, outside. Inside, we have... Okay, let's write down sodium, chloride, calcium, potassium. We have more inside. Let's not do this way. Okay. Cannot erase. Oh. Okay. We have a lot of potassium inside the cell. Let's erase this. We have high concentration of sodium inside, and we have low concentration of potassium inside. We have low concentration of potassium outside, and it's opposite for the other three. We have low inside, low inside, low inside, high outside, high outside, high outside for sodium, chloride, and calcium. All right. Now, what do we mean by this? We definitely see that inside we have less sodium, outside we have more sodium. And do you think this establishes some sort of gradient? Yes, it does. So there is this gradient, right? And this is also called sidedness of the membrane, meaning we have different amount in different proportion on either side of the membrane. Okay, let's talk about this a little more. So obviously there exists a gradient across the plasma membrane for each of these ions. Now let's say somehow the membrane becomes permeable to each of these ions. What will happen? Well, let's draw out a cell and let's draw a channel for sodium. We know we have low sodium inside. Let's say we have 100 sodium inside and we have 10,000 sodium outside. So if this channel is permeable to sodium, and if the channel opens up, what will happen to sodium? Which way will sodium move? Well, understanding the concept of diffusion, we know that sodium will move from outside to inside, right? And it'll keep moving unless it reaches equilibrium. So it'll move from high to low to unless it high to low and until it reaches equilibrium, right? Now the question is, what happens at equilibrium? Will there be still movement of ions when it's at equilibrium? Well, we understand that sodium will follow law of diffusion, and when it comes at equilibrium, we say that there is no net movement. However, individual ions will move from intracellular to extracellular environment and extracellular to intracellular environment and vice versa. But at that point, we say there is no net movement. Now, one thing that students often get confused about is permeability. So let's say I have a cell here, right? And we have high concentration of let's say, I don't know what, let's say some molecules. Okay, we have some molecules, very, very high concentration of those outside the cell, and inside we have really low concentration of the same molecule. So there does exist a gradient, right? We do see a gradient, high outside, low inside. But the main factor here is permeability. If the cell is not permeable to this molecule, then there is no point of having the gradient, because the molecules will not enter the cell in the first place, right? So permeability is a very, very important factor. We have to understand whether the cell is permeable to the particular molecule. No permeability, no diffusion. Good? All right. So let's analyze the chart one more time. It's the same chart that we have been looking at in the last two slides. Let's analyze. So we understand that we have low concentration of sodium inside the cell and high concentration of sodium outside. Now let's say all of a sudden the membrane becomes very, very permeable to sodium, or the sodium channel opens up. Which way will sodium move? If you say it'll move from the extracellular environment to intracellular environment, you are right. We will have an influx of sodium down the concentration gradient. Similarly, let's say calcium. Which way will calcium move if we have the calcium channels open? Well, calcium will move from extracellular to intracellular space. Good. Which way will chloride move? Well, chloride will move from outside to inside. Good. And how about potassium? If the potassium channels open up, which way will potassium move? Well, it'll flow in the opposite direction. So potassium will move from inside to outside the cell along its concentration gradient. So again, you are responsible for knowing the movement of these ions across the membrane. You don't have to remember the numbers, just know which way will they move if the membrane becomes permeable to each of these ions. So you must remember that sodium, chloride, calcium, lot outside the cell. Potassium is inside the cell. Okay. I'll stop here and then start with a new video.